So the first question is straightforward, dilution streaking. So of course, all of you have different patterns of dilution streaking. But essentially what is required is the main aim of a dilution streak is to get single colonies at the end here. Single colonies. Okay. So if you look at this pattern, first this is the dense streak. You get the bacteria growing, and then the streaking. And you can see here that I take less and less lines. Okay. Here I take 3 lines, 2 lines, 1 line. Because the main aim is to dilute, dilution streaking. So you need to reduce your amount of inoculum so that you get single colonies here. Okay. So everyone will have their own pattern, but the most important is it works for you. Okay, you can try out your own patterns, but remember that the main aim, the objective of a dilution streak is to get single colonies around here. Then here shows the different morphology of bacterial colony. So here you need to know the difference between a colony and a cell. Okay, so these are bacterium cells. Okay, cells. Okay, each of them are bacteria. Here are colonies, colony forming units. So lots of bacteria in one colony. So from here, first thing you can see is mucoid. Okay, it's wet. It looks wet. Mucoid. It's entire, which means the boundary is smooth, round, and it's convex. Okay, so this kind of appearance is very typical of Klebsiella, especially Klebsiella pneumonia. Okay, so for this, it's rough. The surface is rough. The boundary is crenate, and it's flat. Okay, it looks flat. Okay, because it's not convex or anything. Here you cannot tell whether it's transparent, translucent, or opaque, because to observe that you have to raise the plate to the light. Okay, if the light doesn't pass through, it's opaque. If it passes a little, it's translucent. But if you can see through it, then it's transparent. Here, most important characteristic is filamentous, rhizoid. Okay, and the surface is rough. Here it's round. The colony is round. Boundary is entire. And it's pulvinate because it bulges up higher than a normal convex does. Okay. Here shows the label of a microscope. Missing labels of a microscope. These are the ocular lens. So this is usually 10 times. Then you have your revolving nose piece, the lenses. Usually you start at 4, 10, 40, then 100. Okay, so at the most you can get about 1000 times magnification or with oil immersion. Okay, if not, then you're stuck with 400 times, 10 times, and 40 times here. So number 2 is objective lens. These are your lenses, objective lenses, your stage, your condenser, diaphragm. So condenser and diaphragm is to control how much light passes through your sample. So sometimes this helps to enable you to see the bacteria clearer. Okay. So this is the coarse knob and the fine focus knob. So these are photomicrographs after gram staining. So photomicrographs means these are the photographs taken under microscope. Okay, so these are typical appearance of a bacterial cell after gram staining. Okay, so the distribution is random. Okay, even color, the stain, you do not get a very nice even color all the way. Sometimes you get uneven coloring. Okay, but not to the stage of variable gram staining. It's just the staining is not even because sometimes the cells are old, the cell wall does not hold the stain well. Fresh young cells tend to hold the the stain better. That's why uh, fresh colonies will give better staining results, except for spore staining. For spore staining, you know that the bacteria produces endospores when they are at their uh, after log phase, plateau phase. Okay, so these are usually older cells. Okay, so usually I will tell the lab assistant to incubate longer so that they give you older cells when you want to do spore staining. However, like if you are given just a plate, like normal plate with bacteria, with let's say typical 5 millimeter cell diameter, okay, 5 millimeter colony size. So you're asked to do spore staining. So where will you take the cell? You take from the edge or take from the center? For spore staining, where do you think you'll get the best staining results? From the center of the CFU colony or from the edge? Spore staining is better for old cells. So where are the old cells? Center or the edge?
In the center, yeah.
Yeah,
Center.
yeah. Okay, so you can play with this provided the colony is big enough to make a difference. Whereas if you're doing gram staining, then you prefer fresh cells, so you take from the edge. Okay, but most of the time the bacterial colonies are too small to make a difference. Okay, this a point. Uh, okay, so here looks you can see it's gram positive. I wrote here is either cocci or coccobacilli because sometimes in real life situations it's hard to differentiate between cocci and coccobacilli. Okay, so this is a gram-positive cocci. Okay, a bluish tinge there. Here, clear gram-negative bacilli. Okay, it's very small. So even if you put coccobacilli, I will not mark you wrong. So this is gram-negative bacilli because there's no, although it's darker, but it's not bluish. Okay, it's not purplish. Here, photomicrographs showing different bacterial cell morphology. So when we're talking about bacterial cell, uh, the labels like filamentous, rough, round, I mean, all these terms are not applicable for cells. Okay, these are all descriptions of the colony only. Okay, so you have to be able to differentiate between colony and cell. So when you talk about cell, here you can see it's a spirilla, okay, spiral shaped bacteria, long. Okay, so this is typical of slides. Okay, sometimes you only see very few bacteria on the microscope field. Okay? You don't see everything clumped together. So this is But you don't, from here, you cannot say, you cannot tell me whether it's gram positive or gram negative because I didn't tell you what stain we're using. I just want the cell morphology, okay? So here you can say it's tetrad, coccus tetrad form, right? Because it's 4, 4, 4. Okay, so this is uh, typical of micrococcus, okay, micrococcus. Uh, but because you don't know whether it's gram positive or negative, you won't know what bacteria it is. But you can say that it is a cocci in a tetrad form. So this is a cocci chain. So if you remember, most typical is your streptococcus. Here is your coccus cluster. Then this is a bacilli chain. Okay. Okay. Most of the bacillus are in this form. Here, these are photomicrographs after spore staining. So here you can see clearly the spore. Okay. Spore is stained by malachite green, whereas the cells are stained by safranin. You can see it's green. So the difference here is I wanted you to see that the spores here are bigger than the cell diameter. So these are actually bulging. So when you talked about when you want to describe spore staining, the position of the spore and the size of the spore is important. Whether it bulges and whether it's at the terminal, central, okay? These are important descriptions to make after spore staining, okay? So here is after flagella staining, you can see amphitrachus, polar flagella at both ends. Then here you can see lophotrichus, a tuft of flagella at one end. So when you are doing flagella staining is technically quite difficult because sometimes the flagella breaks, so you don't see any flagella. Okay, so it will very rarely be evenly distributed. So you have to take a general overview of your slide. Okay, so from here you can say it's a lophotrichus. You can see this one is clearly peritrichus, but you can see this may not be pure colony, but here you can see it's a peritrichus, and this is monotrichus. Okay? And some of them you can see the flagella is lost. Because flagella staining is difficult, most of the time we determine motility from the agar and also look under the microscope to see whether the bacteria is swimming across the field. Here we are looking at hemolytic patterns on blood agar. Okay, so this one a lot of students, some students got it wrong. So this is blood. Okay, so this is blood agar. So these are the colonies. So you can see here there's clearing. This is the Streptococcus pyogenes. You can see clearing. So the clearing is very distinct. So this is typical of beta hemolysis. Here there's a greenish tinge. So alpha hemolysis. And here there is no clearing at all. So it's gamma hemolysis or non-hemolytic. Okay, so this is actually not a very good term to use, but it's been it's the definition that they use. But you can also use non-hemolytic. I think that describes it better. Here, you can see the different morphology when plating on selective and differential agar. Mannitol salt agar. So there's salt here. Salt here is to uh, select for bacteria that can grow in higher salt. For example, here it's a Staphylococcus. Okay. Then for mannitol, it's Staphylococcus aureus. Okay, so if you can ferment mannitol, you get the yellow colony here. Here, this is McConkey agar. So the differentiating component is lactose. So the bacteria that can ferment lactose will produce acid and produce reddish colonies, reddish pinkish colonies. Okay? So these are the non-lactose fermenters. This is hectone enteric agar. Similarly, lactose as a differentiating component. Here you can see this is a lactose fermenter. These two are non-lactose fermenter because they are not yellowish. However, you can see a blackish tinge here. Okay? Because this media allows you to differentiate H2S producer from non-hydrogen sulfide producer. So hydrogen sulfide producers typical of Salmonella, here maybe Shigella, because hectone enteric agar is used for Salmonella, Shigella, and coliform differentiation. So this one, next we look at some of the biochemical tests, common biochemical tests used. So this is the Hugh-Leifson OF test. So they use uh, a different pH indicator. Usually in my lab, I use a simple Phenol Red OF test. Okay, so it's either red or yellow. So Hugh-Leifson OF test, oxidative, fermentative. So here you want to determine whether the bacteria is oxidative or fermentative. Okay, so the first pair is your control, uninoculated test tubes. So this is your first pair. First pair or your reference pair, so that you can compare whatever difference you see here against this. You can see that there is some kind of oil, a layer of oil. Usually we use paraffin oil, a layer on it, to promote fermentation, reduce the oxygen supply, and promote fermentation processes. Here, both the fermentative and oxidative tube is positive, which means the bacteria is able to grow, ferment the glucose, and cause pH to drop. So this is typical of fermentative. Whereas for this one, you can see that only the oxidate, the one that is without the oil layer, you can see some yellowish tinge here. So this is only oxidative and not fermentative. So this is typical of oxidative reaction. Okay, but here you see it's bluish, right? Okay, because this one, that means there's some change versus the reference uninoculated. That means something is growing. But even though it's growing, it's atypical. It doesn't look normal. So it's because it is not it doesn't utilize the sugar here. So usually the term they use is non-saccharolytic. Okay, it doesn't use the saccharides that are present in the uh, media. Usually when it doesn't use the saccharides, probably it uses maybe the uh, protease peptone component and resulting in a actual increase in pH. That's why you see a bluish tinge here. Okay, so you have to compare always against your reference pair. So most of you miss the part where the sugar doesn't is not being utilized. Also for the same for this one. Okay, this is similar. So this is the one that I usually use, Phenol Red Glucose broth. However, usually when I do this, I'll do this in a pair, one with oil, one without oil. So here, Phenol Red, the glucose broth test. So the pH indicator is phenol red, the sugar is glucose. So always compare against the uninoculated. Here the uninoculated is in the middle. Okay. So what do you see here? Both of these are definitely yellow. That means both of them ferment glucose. However, you can see the gas here, gas formation. So this is Durham tube. Durham tube allows you to capture any gas formation. Okay, but there's no gas formation here. This one doesn't ferment glucose because it doesn't change much from the uninoculated. And this one, the color actually grows deeper. Okay, so it's also non-saccharolytic. It degrades the protein, increases the pH, so it becomes darker. Okay, so that's the differences here. Here we'll look at the MRVP test, the pH change. The main difference between MRVP is methyl red positive organisms ferment glucose and produce acid. End product is acidic. That's why the pH decreases over time. Whereas for Voges Proskauer positive organism, all although they produce acid initially, but most of the end products are neutral. Okay, so actually the pH does not become acidic. Okay, so that's the differences between an MR positive and a VP positive organism. Here is typical of a catalase test. Okay. So a catalase test is actually, the reagent is actually 3% hydrogen peroxide. So you don't, yeah. So 3% hydrogen peroxide, you drop the reagent onto your colony. And if you see bubbling, that means the organism has catalase. It's able to react with the hydrogen peroxide and form oxygen. Here is negative. This is oxidase test. You can see that this is the reagent. So if it's positive, it becomes blue. If it's not, it's like this. So catalase is important. Usually we use it to differentiate between uh, Staphylococcus and Streptococcus. Okay, because Staphylococcus is positive, catalase positive. Streptococcus is catalase negative. Here, oxidase, very important because uh, Enterobacteriaceae uh, are oxidase negative, Pseudomonas Ca oxidase positive, Viburnata Ca oxidase positive. So three very important family. Then for the OF test, similarly, Pseudomonas Ca is oxidative, Enterobacteriaceae and Vibrio Naceae is fermentative. So this shows the results of the Simmons citrate test. Here is the uninoculated, so this is very clear. Here it's positive, here it's negative. Okay, so it's bluish because it's a Simmons citrate test. So the only carbon source is citrate. That means it can break down citrate. Okay, so but when it breaks down citrate, it has when an organism uses carbon, it also has to use the nitrogen to supplement the carbon intake, because most of the molecules that you produce, whether DNA, RNA, it doesn't you don't only use carbon to produce them, but you also need your P, your N, other components, other elements. So so in order for the organism to use the citrate, they also have to break down the ammonium dihydrogen phosphate. So when it does this, the the media pH of the media changes into alkaline, and you changes from green to bluish. This is the result of a decarboxylase test. So here you have to do it in a pair. So this is the uninoculated control. This is the inoculated. So both of them are inoculated. This one, the color is yellow because it only utilizes the uh, sugar, but it couldn't continue utilizing the amino acid. Here, if it continues using the amino acid, it becomes purplish. So usually when they do the amino acid utilization test, usually we do it in pairs, no, in sets of arginine, lysine, and ornithine. Okay? Arginine, lysine, and ornithine. These are the three main ones. Sulphide Indole Motility agar, the SIM test. So for motility agar, the agar is usually about 0.5%. Normal agar on your plate is 1.5 to 2%. So when it's 0.5%, it's very, very soft. It's soft so that it allows the bacteria to swim out. So if the bacteria is motile, it will cloud the whole tube. If the bacteria is not motile, it will only grow along the step thread. Okay, like this. So on this is a very beautiful picture, but in actual work, in actual lab, it is very, very difficult to step steadily because most of the time your hands will shake and you will get a very, very big uh, step. So this test requires practice. That's why sometimes I will just use the fresh culture, look under the microscope to see whether it's moving, instead of doing this, because it's not easy to get a nice step. Okay, so both are not producing sulfide. You don't see any blackening here. Okay, you can see that the colony is actually growing on top. Okay, it's spread all over already. So this is the TSI, triple sugar iron agar. So triple sugar iron agar, whenever you describe triple sugar iron agar, you must know that there's three types of sugar: glucose, sucrose, and lactose. Okay. So here you can see that the bud is yellowish, here is not. So it's because that it only utilizes glucose, not enough to, the fermentation of glucose is not enough to maintain the whole tube yellow. Okay. So if you incubate it overnight, the proteins catabolize aerobically and on a slant will change this to revert this back to pinkish. So this is typical of only glucose fermentation. This is the uninoculated control. Here you can see there is some reddish here. So you see protein catabolize aerobically, but nothing happening in the bud. So this is slant, this is bud. Here, definitely you can see hydrogen peroxide, a hydrogen sulfide formation. You can also see that there is a alkaline slant because it's reddish here, right? Here, all the sugars are utilized, very yellow, and a lot of gas formation. That's why it broke. This is the Bacitracin susceptibility test. Just to usually this is how you also carry out your antibiotic susceptibility test, where you put the antibiotic disc onto a lawn of a bacteria. So if you see a clearing zone, that means the bacteria is sensitive. If not, that means the bacteria is resistant.
No.
Okay, so this is the coagulase test. So here you can see coagulase positive, here is coagulase negative. So coagulase positive is when you see the cells clumping up together. Okay. The because so this is usually used for to differentiate between Staph aureus, coagulase positive Staph, and coagulase negative Staph, usually Staphylococcus epidermidis.